Felicity, what do you call an acid with an attitude? Amino acid.
Oh my. Ari, why did the white bear dissolve in water?
I don't know. Why?
Polar bear.
So, the paper we are presenting today is about structural and biochemical characterization of SADS-CoV, the papain-like protease, by the authors shown there. And this presentation will consist of introduction, methods, results, and discussion. SADS-CoV is a coronavirus from the alpha coronavirus family. So it is also stemming from bats as COVID-19 is. It was a variation found in southern China when it caused the death of 24,693 piglets in 2017. So, due to numerous alterations in the coronavirus family, it is very difficult to develop a versatile antiviral, and there are no current vaccines against this novel virus. So, this research paper focuses on a papain-like protease, PLP, that SADS-CoV encodes, because this PLP promotes viral replication in the host by cleaving polyproteins. The two polyproteins it cleaves are ubiquitin and ISG15. So it displays deubiquitinating and deISGylating activities that prevent ubiquitin and ISG15 from stopping viral spread. This experiment provides more information about Plp2 to develop an effective antiviral. So the first method to isolate Plp2 was affinity chromatography. The supernatant cell residues were added to the column in a stationary phase, and the lysis buffer went through to open the cells. Then a wash buffer was sent through to get all the proteins that were not needed and elute them. And another elution buffer was sent through to remove the purified PLP2, which was then mixed with a ligand, which was the sumo tag, in a known concentration of 1 to 200. Then affinity chromatography was used again to remove the sumo tag from the PLP2. And finally, the PLP2 was sent through a gel filtration buffer to be purified, and after being purified, it was crystallized by a sitting drop vapor diffusion. Another method used in the experiment was site-directed mutagenesis, which makes specific and intentional changes to the DNA of a gene, specifically of BL21D3, which are competent bacterial cells, and these cells are commonly known for protein expression as they are high efficient. So they were the ones producing the PLP2. Another method was ubiquitin ISG15 AMC and peptide AMC kinetics. So there were 4 different fluorescent substrates used to determine a catalyzed reaction rate of SADS-CoV. They found out that as the fluorescent increased per minute, the amount of AMC released also increased. So this was kind of, it had a linear relation, and the initial velocity of this AMC released was taken to find the steady state enzyme kinetic parameters.
Okay, so now we can get into some of the results from the experiment. And the first result was from their test of SADS-CoV PLP2's catalytic efficiency. So they tested for its cleaving efficiency by mixing PLP2 with NSP1 EGFP, and NSP1 is a non-structural protein, which was bound to an enhanced green fluorescent protein. And they were just testing its ability to cleave this complex, analyzed by SCS Page, which is sodium dodecyl sulfate polyacrylamide gel electrophoresis. It's a technique commonly used to characterize proteins by molecular weight. But in this case, we were just trying to see if PLP2 could cleave the complex and create two smaller NSP1 and EGFP proteins. And this in SCS Page, the smaller proteins migrate further toward the positive charge at the bottom, and the original NSP1 complex should be larger and heavier and not travel as far down in SDS Page. So the results were that PLP2 successfully cleaved the complex to create the free proteins, and that's clearly seen by the two lines at the bottom of the SDS page. The other results involved the overall structure of the PLP2. So its structure kind of resembles your left hand facing towards you. That's the best way to visualize it. And it consists of the thumb domain in pink there, and the fingers domain in yellow, and the palm domain in blue. And the overall PLP2 structure is comprised of 293 amino acids, and the thumb domain consists of 6 alpha helices, a 310 helix, and 2 beta strands arranged as a beta hairpin structure, where it contacts the palm domain, which consists of 7 beta strands. And then finally, the fingers domain has 1 alpha helix, 4 beta strands, and 1 310 helix. There's also that zinc ion, as seen in gray, and it is coordinated in the fingers region by 3 cysteine residues and 1 histidine residue, and it kind of resembles a ring on one of the fingers. So zooming in on figure B from the last slide, we can see a really nice structural comparison of all of the cleaving portions of 4 different coronaviruses. To start, our SADS-CoV is in yellow, and the blue, which is SARS-CoV, and green MERS-CoV, both deviated in the fingers and palm domains slightly from SADS, whereas TGEV-CoV was much shorter, and you can kind of see it in pink. It stops right before the thumb domain, so it's probably considered the most structurally different from SADS-CoV. And on the left, you can see the primary structure, which is the overall amino acid sequence comparison. And the red blocked backgrounds are exact matches of amino acid sequence across all 4 coronaviruses. And then the ones with just red letters have some similarities, but not all 4. And then any black lettering is just differences. So even though they're all in the same coronavirus genome, there's actually very little similarities in their cleaving mechanisms. And we can see later that that makes a vaccine really hard to achieve. PLP2 also, it causes the deubiquitinating and deisgelating activity by binding to its specific substrate, which in this case is ubiquitin. So it has a unique binding site to promote this replication, and it's really difficult to observe how it binds to ubiquitin because it can never be crystallized in that intermediate complex. They can only crystallize the reactants or the products afterwards, like we saw in the SDS page. So this is an area where more research is being done. Well, SADS-CoV PLP2 binds to ubiquitin by forming a salt bridge with a side chain of glutamine 168 and the side chain arginine 42 of ubiquitin. Okay, so the results of this study were kind of vague, obviously, because there's still no vaccine achieved for a majority of the coronaviruses, and COVID-19 included. So the results showed that SADS-CoV PLP2 successfully cleaved the non-structural protein 1 and EGFP as seen in the SDS page. So that's how it replicates itself and promotes virus spread. The overall structure was similar to SARS, MERS, and TGEV-CoV, but they did display slight variations, especially in their active sites where they bound to their specific substrate, and then SADS-CoV specifically binding to ubiquitin. They also tested its binding affinity to ZKAGG-AMC and ZLRGG-AMC, which their structures are shown to the right. Results showed that SADS-CoV binded better to the ZKAGG-AMC.
So this study provided much needed structural and biochemical characterization of PLP2 specifically, since it helped us understand better the deubiquitinating mechanism, and this helped us understand how it interacts with other substrates better. But a vaccine is still hard to design, as there are many variations between coronaviruses. It will take more research to find a successful antiviral that can be produced.
Thank you.